Molecular identification of microorganisms. So target microorganisms for the molecular-based testing are those that are difficult or time-consuming to isolate, for example, mycobacteria. Hazardous organisms like Histoplasma coccidioides, those without reliable testing methods, HIV, hepatitis C, and high-volume tests, for example, Strep pyogenes, Neisseria gonorrhoeae, and C. trachomatis, or Chlamydia trachomatis. Now, Chlamydia trachomatis can't grow outside of human cells, so it would be impossible to culture it. It's different from like gonorrhea, where gonorrhea we could culture on chocolate agar or MTM. Chlamydia can't grow in routine agar. It needs cells to grow inside of. So chlamydia is a good one that we would use for nucleic acid amplification testing or NAT testing. Molecular testing is becoming much more common in clinical microbiology due to the turnaround time. So oftentimes, patient comes in with an infection and the doctor wants to be able to treat the patient almost immediately. So many of our molecular tests today are being designed as like multiplexed tests where they'll do like a body region and think of the most common organisms associated with that body region and test for all of those at once. Now, multiplex PCR's assays are still being developed, so they aren't readily available, but there are some that are out there that are commonly used. There's one that's associated with like blood cultures, because getting the identification of which organism is causing sepsis is really important. Sepsis can kill people. So, those types of tests are becoming increasingly more popular. It's estimated that pretty soon, routine culture methods are no longer going to exist in the clinical microbiology lab. The clinical microbiology lab will be converted to molecular testing. Now, there's of course some benefits that still come with diagnosing the traditional way. So being able to culture the organism and identify which organism is causing the infection, especially if it's a mixed infection, if there's multiple organisms within there. Sometimes multiplex PCRs can't identify everything that's in there, and we miss key features of the entire picture if we don't actually grow it on a plate and look at the different organisms that grow on the plate. But if we can't grow it, if it's really dangerous to grow, or if there's other confounding issues, then using molecular testing is important. The other aspect for testing microorganisms for molecular-based testing or targeting microorganisms is to look for antibiotic resistance genes. Now, it's really important to note that if a microorganism has a gene that would confer a protein that allows that microorganism to be resistant to a drug, it doesn't mean that it will actually produce that protein. The gene has to be able to be turned on at some point. So just because the gene is present, it doesn't mean that it is phenotypically expressed. So throughout this next few lectures that we'll go through, we'll talk about the difference between genotypic presence and phenotypic expression. So just up front here, genotypic presence means that it's present in the genome of the organism. So the genes are there. Phenotypic expression means that the genes were actually turned on, transcripted, and translated into the protein. And the protein that was produced was functional. And it was produced to the extent that's required to show the resistance. So in this case, we're only talking about antibiotic resistance, but later on we'll talk about cancer and we'll talk about, different types of genetically inherited diseases. So it's important to note that just because the genes are present doesn't mean that the genes are going to turn into a functional protein. And even if they turn into a functional protein, is it going to be enough to actually start to have disease symptoms? All right, so applications: rapid or high throughput identification of microorganisms, detection and analysis of resistance genes, genotyping, classification, and the discovery of new microorganisms. Specimen collection: so you want to preserve viability and nucleic acid integrity of the target microorganisms. Avoid contamination. Appropriate time and site of the collection: blood, urine, other. Use proper equipment: coagulant, wood or plastic swab shafts. Commercial collection kits are available. Consider the specimen type, for example, stool, plasma, cerebrospinal fluid. More rigorous lysis procedures are required to penetrate cell walls. So cell walls are different from doing an extraction on human cells. If you want to be able to get through the cell walls, you need to have a pretty rigorous lysis procedure to be able to break open bacterial cell walls, or even like yeast cell walls, any type of fungal cell walls. Those are going to be a lot more rigid and difficult to penetrate, so that has to be kept in mind as well. Consider the number of organisms in the sample. Inactivate inhibitors, acidic polysaccharides in sputum or polymerase inhibitors in CSF. Inactivate RNases. Target selection. So here's an example of where we have the target organism, the variant of the target organism, and other normal flora. So all of the microorganisms have region A in their genome. So whether it's the target, the variant, or normal flora, they all have this. We would target 2 additional areas. One additional area would be just specific for the target organism. So the variant and the other flora would not have it. The other area that we would target would pick up the variant. So it would pick up the initial region, plus it would pick up the variant. So this would capture not only the target, but also the variant, while remaining negative for a normal flora. PCR and other amplification methods are extremely sensitive and very specific. For accurate test interpretation, use proper controls. So a positive control would contain the positive template. A negative control would contain the negative template. And an amplification control is omnipresent. So it's always present. It confirms that the specimen contained human sample. And it confirms that the amplification system is working properly. The template is unrelated to the target, reagent blank, no template present. Internal controls, so homologous extrinsic is added known target sequence. It controls for the amplification process. Heterologous extrinsic is added target that's different from the target in mind. So it's an added sequence that is different from the target that we're looking for. It controls for the extraction and amplification procedures. And heterologous intrinsic is a human gene control. So that's a gene that we're looking for from within a human sample. Contamination, you want to check the reagent blank. Dead or dying organisms, you need to retest 3 to 6 weeks after antimicrobial therapy. Detection of less than clinically significant levels. So those would be false positives. False negatives, improper collection, specimen handling, extraction, amplification failure, check internal controls. Technical difficulties with chemistry or instrumentation, check the method and calibrations. So just going back here, a false positive, as you know, is a positive result, but it should be negative. False positives can be caused by contamination, dead or dying organisms, and the detection of less than clinically significant levels. False negatives would be a negative result that should actually be positive. If we didn't handle the specimen correctly, maybe we destroyed all the genetic material. Extraction or amplification failure, maybe the controls did not work, the system wasn't working, so it wasn't able to detect anything. Even the positive control ended up being negative. And technical difficulties with chemistry or instrumentation. Check the method and calibrations. Some of the procedures are very touchy, and adjusting, like PCR, for example, by 1 degree will impact the recoverability of organisms. So if we have those settings incorrect, then we would get a negative result, and maybe it's not negative. Viruses, the classic methods of detection include antibody detection, antigen detection, and culture. Molecular methods of detection include target, probe, and signal amplification. Tests are designed for identification of viruses, the determination of viral load, or the number of viruses per milliliter of fluid, and genotyping by sequence analysis. The viral load is the number of viruses per milliliter of body fluid. It's measured by quantitative polymerase chain reaction, or qPCR, digital PCR, or other methods. Measurements by different methods may not be equivalent due to technical variability and biological variability. Test performance features for the viral load measurement: sensitivity, the lowest level detected at least 95% of the time. Accuracy, the ability to determine the true value. Precision, the reproducibility of the independent determined test results. Specificity, negative samples are always negative, and positive results are true positives. Linearity, a serial dilution of standard curve closely approximates a straight line. And flexibility, the accuracy of measurement of virus regardless of sequence variations. So if there's slight mutations in the sequence, we could still accurately detect it. So the system allows for some flexibility in detecting the wide range even if mutations are present. Antimicrobial agents, so if they inhibit growth, it's static, for example, bacteriostatic, fungostatic. If they kill the organisms, it's cidal, for example, bactericidal, fungicidal, virucidal. Antimicrobial agents are classified by static or cidal, the mode of action, and the chemical structure. So these are sites of action of antimicrobial agents, and this should be a review. Mechanisms for the development of resistance to antimicrobial agents, enzymatic inactivation of the agent, altered target, altered transport of the agent in or out, and acquisition of genetic factors from other resistant organisms. Viral genotyping. Viral genes mutate to overcome antiviral agents. Gene mutations are detected by sequencing. Primary resistance mutations affect drug sensitivity but may slow viral growth. Secondary resistance mechanism or mutations compensate for the primary resistance growth defects. Advantages of molecular detection of resistance to antimicrobial agents. Mutated genes are strong evidence of resistance. Rapid detection without culturing, and direct comparison of multiple isolates in epidemiological investigations. A normal gene may be mutated, which makes it resistant to a drug, or the microorganism got a gene, an extra gene, usually on a plasmid, that confers resistance. For example, the MEK-A gene confers methicillin resistance. Acquiring that gene makes Staphylococcus aureus resistant to methicillin. Molecular epidemiology. So sometimes we want to track diseases across countries or across different parts of the world. An epidemic is a rapidly spreading outbreak of an infectious disease. A pandemic is a disease that sweeps across wide geographical areas, usually multiple continents. Epidemiology is the collection and analysis of environmental, microbiological, and clinical data. Phenotypic analysis measures biological characteristics of organisms. Molecular epidemiology is a genotypic analysis targeting a genome or plasmid DNA. Species, strain, or type-specific DNA sequences are the sources of genotype information. So when we do the criteria for PFGE pattern interpretation, we use the rule of 3. So if they're indistinguishable, there's 0 genetic differences and 0 fragment differences. Closely related are listed, possibly related and different are listed here. And then this would be a PFGE that we would run out to make that determination. Arbitrarily primed PCR, or random amplification of polymorphic DNA, RAPD taxonomic studies. So here we have arbitrary primers. So they randomly amplify different regions. We don't set them up to amplify a particular region based on a known sequence. We just say, here's a forward primer, here's a reverse primer, go ahead and amplify anything you can. Now, if the organisms are closely related, then when it's randomly amplified in various regions with all these different primers, if they're closely related, you're going to have a lot of the similar bands. If they're not related at all, then all the bands are going to be pretty different. And this is used for taxonomic studies. Interspersed repetitive elements, like DNA fingerprint or create DNA fingerprints. Interspersed repetitive elements are repetitive elements that are interspersed amongst the genome. PCR amplification priming outward from repetitive elements generates strain-specific products. So then you can differentiate strains based on those repetitive elements. Other genotypic methods used to type organisms: plasmid fingerprinting with restriction enzymes. So we cut up the plasmids and then run them out on a gel and figure out the different bands that are produced. RFLP analysis. So instead of just looking at the plasmid, we do restriction fragment length polymorphism of the genome. Amplified fragment length polymorphism. We amplify a particular region and then evaluate the length differences. Interspersed repetitive elements we just talked about. Ribotyping. That's usually evaluating the ribosomal RNA. SPA typing, multi-locus sequence typing. And here you can see the typing capacity, discriminatory power, reproducibility, ease of use, and ease of interpretation for the various tests. As I mentioned in the beginning, multiplex PCR by body site are becoming available. So this is one kit that shows one kit for 6 bacterial detection based on the body regions. So it has Klebsiella pneumoniae, Streptococcus pneumoniae, Haemophilus influenza, Pseudomonas aeruginosa, Legionella pneumophilia, and Staphylococcus aureus. And there's different kits that are available for different categories of microorganisms. And then you can go ahead and review the summary.